The, this is a webinar format, so that means that participants do not have access to the microphone nor to the camera, but if you have questions, you can ask them in the Q&A, and the head of the school, Ana Ituriaga, will be happy to propose them to Mr. Pao. And if you have comments, you can put them in the chat, and we will have a look at this as well. Have a good conference, and Ana, the floor is yours.
Thank you so much, Helen, and thank you, of course, to Gerald for being with us once again. And welcome to everybody who's listening to another one of our user online talks. You know, this is our space where we find top speakers to make us reflect about political, economical, societal, health issues that are making the headlines today. And as you may know, we have had almost 300 talks since the start of the confinement on every single topic under the sun. But if you missed one, no worries. You can always go to our website, and I invite you to do so because I am sure that you will find something that tickles your fancy. So please go in there and see what we have for you. Another one of our objectives is to inform and learn about the issue with capitals of the moment. So the issue in the headlines yesterday, today, and probably many months to come, and that is COVID. And for that, we have been connecting over the past months with top scientists across the world to understand more about the virus, its behavior, the spread of the pandemic, and how are different countries managing the crisis. Because the appearance of the COVID-19, or in its full scientific name, SARS-CoV-2 virus in late 2019 has led to the upheaval of the global order in ways unimagined before. It is a virus infecting human cells and replicating itself very rapidly, spreading infection and having affected already 5 million people. We have also seen the apparition of different variants and mutations from the original Wuhan virus to some others coming from the UK, from India, the famous Delta variant, and now also from South Africa with the Omicron mutation. In a previous session, Gerald looked at the latest understanding of science on the virus, on testing, on vaccines, and he also explored scientific efforts that are ongoing to overcome the pandemic. And now that we're hearing everywhere about the newest variant, this Omicron that I just mentioned, Gerald has kindly accepted to come back once again to let us know a bit more about these new variants. Because given its great capacity to evade the immunological system and to spread fast, it seems almost 3 times faster than the Delta variant, it may become soon the dominant one. And there are new fears and question marks that are emerging, like, is it true that this variant is more contagious but less virulent? Can it be neutralized with existing vaccines? And what about treatment with antivirals like Paxlovid or Molnupiravir? We're confident that Gerald can help us answer some of these questions. So Gerald, once again, thanks so much for being with us and giving us updated, rigorous information on the COVID and its new variants. But before I give you the floor, I would like to say a few words about you in case our colleagues have not heard it before. You are a scientist at the Salk Institute in La Jolla, California. You grew up in Spain with a German education and studied molecular biology at the University of San Diego. And then you went on to medical school at the University of Washington and completed your PhD at the Salk Institute. You conducted postdoctoral training at the Salk Institute in the Laboratory of Genetics and at the Climate, Atmospheric Sciences and Physical Oceanography. Your work spans from molecular evolution, stem cells, cancer, biology, neuroscience, chaos theory, and has over 20 years, and you have over 20 years experience in virology and viral engineering. In February 2020, after realizing the threat of the new coronavirus, you and a small group of scientists in California set out to generate research tools to enable virology research on the COVID. Your current efforts are centered around a monitoring and early warning system of SARS-CoV-2 variants that could threaten the effectiveness of the vaccines. So, as I said, thank you so much, Gerald, and the floor is yours.
Thank you for having me. Well, I'll try to update everybody here with up-to-date information on SARS-CoV-2. You know, bear in mind that all of this is a quite fluid situation and some of the things are not as certain. And when they're not as certain, I will mention to you where these uncertainties lie. But this is the, you know, as science itself, this is basically, ongoing update based on the best available evidence. Okay, so just to recapitulate here, let me see. So this is, as you may all have seen this before, but maybe not in such detail. This is the, change this a little bit because I have okay. So this is the genome of the SARS coronavirus 2. And that genome is basically about 30,000 base pairs, which is like 30,000, what you call 30 kilobases. But of all the genes that you actually have here, the ones that we care mostly about are the spike, envelope, membrane, and nucleocapsid. And this is basically, and we call it SEMNN. And one that we mostly care about most of the time is the spike protein. Why? Because the spike protein is on the surface of the virus. And this is sort of like the business end of the virus. This is like how the virus interacts with the world when it's out there. And and this is basically what we want to have like the immune response work against, because this is what's exposed. If you were to immunize somebody and you have to block things, you will have to block through the spike protein. So the other viruses, the other proteins and the other genes are important for the virus life cycle, but they're not part of the physical particle itself. Basically, they have this designation NSP, which is like non-structural protein, whereas this SEMNN are the so-called structural proteins. Okay. So as I mentioned, that the spike protein is where the interest is. That's basically, that is what the business end of the virus is. And the virus actually has to enter the cells, and it has to, when it enters the cells, is through this ACE2 protein, through which it enters, but the spike protein itself has to be processed. And there's this protein called TMPRSS2, which is a protease. It's a protein that cuts other proteins. And that is basically has to do 2 cuts on the spike protein in order to activate it. So that's something necessary. And this is basically, this is like a more molecular detail, the overall structure of it. And actually you can see that this is the human ACE2 protein, and this is the so-called receptor binding domain. This is how it interacts with the cell and then actually goes in. So that's why we care about this area. And as you can see here highlighted are all the mutations that exist on the Omicron variant. So you can see a lot of different ones. And the most important regions for the immune response are here in this region. And there's like this NTD, there's called N-terminal domain, as well as the receptor binding domain. And those are basically 2 of the 3 surfaces that are important for viral neutralization by antibodies. So antibodies will neutralize it by covering up. There are other proteins that bind up and, for example, compete with the receptor itself. So if you block this interaction surface with the cell, the virus cannot enter the cell and cannot infect. So then the immune response essentially does that. So over time, as we have seen, and since sort of like the end of 2020, when the Alpha variant came up, the variants tend to be completely dominant wherever they show up. So basically, like this is basically the over time how the Alpha variant increased. But then when the Delta variant appeared, it basically displaced the Alpha variant and became almost complete. And then what you can see here in red here, what you see here, this is the appearance of the Omicron variant just in the last few weeks. This was first detected in South Africa. And, just on American Thanksgiving, the South African health authorities showed that there was appearance of a new variant that was spreading very fast and was able to displace Delta. So the epidemic in South Africa was a little bit different. Unlike in Europe and most of the world where Alpha was dominant, they had their own variant called Beta. And Beta displaced everything. A Beta had the capability of being more immunoevasive. And initially, we thought that beta was going to be dominant and going to threaten the vaccine. However, beta did not go beyond South Africa. And the reason we thought that it didn't go beyond South Africa is because there was a high level of infection, a natural infection of the virus. And in South Africa, when people were previously infected, it was very immunodominant, meaning that it could compete with the other virus that existed previously, the Wuhan virus. And therefore the beta was successful there, but probably not in the rest of the world where only Delta was successful. But once Delta got into South Africa, it was able to displace beta almost completely. And now it seems that this is B11529, which was the original name of Omicron, is starting to displace Delta at a very high rate. And this was like a release by Tulio de Oliveira, who is basically a bioinformatician that works with the South African authorities. And they have done an excellent job in monitoring the situation in Africa. Actually, they are the best monitoring system that exists in South Africa and in Africa in general. So we are very grateful to them to have found this, because they actually released this data when they had just 77 cases in the entire country. And then this only appeared sort of at the very beginning of November. By the end of November, they were able to tell the rest of the world that this was a serious threat. So why do we care? There's so many other variants, and why do we actually care about, Omicron or B11529, which is its original name, more than the other ones? Well, first, this was also from the original release of the information on, which was on November 25th or 24th. And basically, so when they actually see the mutations on spike have the potential for resistance to neutralizing antibodies. So basically they can actually, the mutations that happen is basically you have changed particular amino acids on the spike protein. When you change those amino acids, the antibodies are not able to bind. So an antibody typically binds a stretch of 8 amino acids, but these amino acids are not random. There are certain ones that are nicely exposed on the surface that tend to be preferred. And those are the ones that work well. So if you actually change the amino acids, they're no longer what the antibody recognizes, so therefore it can escape. So the mutations they saw are in regions also that seem to increase the cell entry, which also means that the cell, basically the spike becomes more efficient, and therefore it would lead to enhanced transmissibility. And in other genes also, they saw certain mutations that indicate that is immune evasive. So it basically like it is. There are other types of the immune system called innate immunity, and there's something called interferon, and the mutations that are there seem to also indicate that it enhanced transmission through suppression of the innate immune system. And there are other mutations that we don't know how they actually work. For example, in the nucleocapsid, in the N protein, that we know increases infectivity, but we don't know exactly how. So we saw this constellation of mutations happening in Omicron, and also that fast transmission combined seem to have fueled the suspicion that this is actually something quite different, and therefore we should pay special attention to. So one thing is, like, if we look at the dominant variants that have happened over time since the original Wuhan strain, Alpha had a few mutations and Delta had some mutations, but actually less than Alpha, but became much more dominant. But Omicron has a ton of mutations, actually 32 to be exact, on the spike protein. And since I told you that this is the business end of the spike protein, this is why we worried about it. As you can see, Omicron also has some other mutations. It has a total of 51 or 52 mutations. And now we call this lineage BA1. The names keep changing, although it was originally B11529, you can see the vast majority of mutations of Omicron are on the spike gene. Actually 32 out of the 51 mutations. If you compare Delta, these are the mutations on Delta compared to the original Wuhan strain. You actually have many, many more. All these 32 mutations are mapping on these important parts, especially here, which is the receptor binding domain and the so-called N-terminal domain. That is basically where we actually saw immune escape. So some of these immune escape mutations are there, and also the ones that enhance transmissibility. As I showed this before, these are the immune escape ones through the receptor binding domain, this N-terminal domain, and also around the cleavage side, which are the important ways. And then you can actually see on the right where the mutations are and basically how this actually looks in 3D. So when we actually have here like things to say like a likely adaptive, potentially adaptive, unknown adaptive value, means that they are evolutionary changes that are benefiting the virus somehow, and that the ones that we are quite certain, the ones that make sense that it would do something, and the other ones that we see that are in worrying regions, but we actually don't know how they actually work because we don't have data on that. They're completely new, for example, and then we don't know exactly just because of the place where they are or because of the type of changes that are change things a lot, chemically speaking. Those we suspect them to be adaptive. Sometimes there is basically statistical things. For example, that is that if you have a change in the if you have a mutation that changes an amino acid that basically, and you don't have corresponding so-called silent mutations, you actually have that we call evidence for positive selection. That is basically evolutionary pressure to go in the direction of changing something that's probably beneficial for the virus somehow. So if we look at the evolutionary tree, it's like delta. This is basically delta that was like dominant, and then you can actually see the appearance of Omicron here. So it basically like came a little bit without any relatives, and it appeared like from a very, very deep branch, and then by the time that we found it, it's basically it expanded here. So, but it probably came out from something that is quite different. And if we look at the evolution, it has basically a somewhat of a mysterious origin. And here, this is basically emphasizes how this is a little bit unusual. If you actually look at this, this is basically if we count the number of mutations from the original Wuhan strain over time and the different viruses that we have sequenced over time, they follow this line more or less. And if they're above this line, this is they have more than your average number of mutations. It's like it's like below this line, it has like less than the average number of mutations compared to the Wuhan strain. And you can see Omicron is basically on the high side if you look at the whole viral genome, but it's not terribly a whole lot more. And based on this thing, because over time you actually accumulate mutations, and the number that we actually have, we can infer when this Omicron appeared. And then if you look at this, so it must have appeared around October or something, early the end of September, early October, somewhere between September 9th and October 13th is your basically 95 confidence interval. So this is within this time, probably center around September 30th. But what people worry most is because if you actually do the same thing, but just for a fraction of the genome, basically specifically the fraction of the gene part of the spike protein, if you look at the number, you can apply the clock not to the whole virus, but apply the clock to just the S1 portion of the spike protein. There, then Omicron looks quite different. You can see this is the line for everything, and then it has an unusual number of mutations in there. So this suggests that it's highly adaptive. It has adapted. It has added many more mutations that it statistically would actually have if it were just running a normal clock. So it has an accelerated clock, and it accelerated towards an objective. And the objective is to spread better or to escape the immune system better or something along those lines. So that led us to consider two possibilities. And the possibilities is basically that, and also we did not see any other viruses in between. So you find Omicron here, but you find no other viruses, along this line towards Omicron. So this could be, due to lack of surveillance. So due to lack of surveillance, how can it actually happen that we have this lack of surveillance? The South African authorities are pretty good about sequencing the material. They have a good infrastructure to do so. And we actually had seen this before with the Alpha variant. The Alpha variant also had basically an unusual number of mutations. And there in the UK, about 5%. So every 20th case in the entire population is actually being sequenced. So they have a really good surveillance, but we saw nothing in between. So this must have been hiding somewhere. So since we know it has been hiding somewhere, there can be like two possibilities. One, there was a single immunocompromised human, for example, that is basically chronically infected for a long time. And we actually saw that whenever we gave antibodies to a particular patient, that, there's one of the mutations that appeared in the N-terminal domain, that's like Delta 6970. That mutation appeared whenever people gave, antibody treatment to the patient. That mutation started to appear. When you take the treatment away, it disappears. When you treat them again with antibodies, this mutation appears. So we think that this escapes the antibody treatment. So like in this way, immunocompromised humans can be like allow the virus to mutate more and many times and have a slow immune selection, not enough to suppress the virus. So you allow the virus to accumulate mutations over time. And since this is just one person, the chances of you finding it by immunosurveillance, by surveillance, by sequencing is very unlikely. The other possibility is that there's a human, or we know that the virus probably came from humans where it's mostly widespread. Then it went to an animal, and then it went back to a human again. So it's a spillover onto an animal, and then a second zoonosis. The animal just spreads it back to a human, and then and because of the spectrum of mutations that we actually see on Omicron, we think that maybe the possibility number two is like is more likely, because some of these mutations that we see in the spectrum of mutations appear to be things that would be favorable if the virus was actually in a non-human host. So that's why we think that, it's maybe more likely for this have been a human to animal to human transmission. Whereas in Alpha, it seems to have been an immunocompromised human only that spilled back into the community. If you look at, so this is what worried people the most. So you saw that if you actually compare, in South Africa, you had like 3 waves prior. You had the first original Wuhan wave, then you have the beta wave, and then you had the Delta wave. If you compare the number of daily cases, the test positivity that you actually have, and also weekly hospital admissions, if you compare these 3 things, you see Omicron is growing much faster and at a much steeper rate than any of the previous ones. So this is the worrying part, that is actually accelerating. And then this also happens with every measure. So basically with the daily cases, with the positivity, but with hospital admissions, it seems to track all the other ones. So some people have speculated that this is due to less severe infection, but it's equally likely that this is because of a partial immunity that pre-exists. So people were already infected to a large extent. Most people in South Africa in the population were probably infected by some form of SARS-CoV-2 and had COVID before. So they had some partial immunity to it. And this immunity might have given some protection. So with anything subsequent, maybe the infection is going to be milder because you have partial protection, but not sufficient to fend off this virus, but sufficient not to, end up in the hospital as easily. One thing that's interesting is from the hospital reports that we get from South Africa is that there is a shift towards younger patients. So the people who are ending up in the hospital are frequently people under 50, not over 50. And also there is there's a preponderance of, college students and school children, as has been reported. So here I'm going to show some information from the UK Health Security Agency, which has also done a superb work. And if you actually compare in South Africa, this is another reason why we have. So if you actually check Delta, this is the number of cases, and this is logarithmic scale. So every time that you have a notch here, it goes up by 10-fold. So, so if it has an exponential growth, will show up as a straight line. So you can see that in South Africa, Omicron has been quickly declining and have kind of fluctuating in levels. But suddenly when Omicron appeared, this is going straight up. In the United Kingdom, Delta was staying at a steady level. And then you see Omicron when it's being introduced, in late November, it just going straight up. So this is like, this means that Omicron is displacing. And in the South African setting, the doubling rate that you actually saw is basically 0.21 per day. So the doubling time is basically roughly 4 to 5 days. And in the United Kingdom, the doubling rate is every 2 days. So this is, I want to just point out, in particular settings, the doubling rate for the virus is not a universal property. It depends on its environment. So the environment in South Africa is having you doubling rates at, at 4 days or 5 days. But in the UK, you might have a doubling rate of 2 days or maybe less. And if you actually look at their data, if they compare all the other or the appearance of all the other lines, basically all the other viral variants, you can see Omicron is faster than anybody else. So it's spreading way faster. And here, their latest estimate was that basically the growth rate was 0.35 per day. But, the reports vary between 2 and 5 days. So here, basically, you can see that. So we expect that every 3 days, the number of cases the new cases that you actually have are doubling. But you have to remember, each of those cases will actually transmit to somebody else, which also transmit at this enhanced rate. So this is like exponential growth. So this is like quite worrying that this is actually growing at this fast pace. So here in this report, it's showing that if we compare Delta and Omicron, Omicron is at least basically this is what you call the secondary attack rate, and that's basically you try to measure how from basically household contacts, how frequently you actually infect other people. And if you compare Omicron and Delta, you can see approximately a 2-fold increase in the UK setting compared to Delta. So it's twice as contagious. But we, depending on different people measuring this in different settings, there has been reported to be up to 8 times more contagious than Delta itself. So, assuming a doubling rate of 2.5 days, by Christmas, we should have more than 1 million cases per day. So this is basically based on their current model. Things might change it or in one way or the other, but this is, we will be back to numbers that are like, so this is like, even we think that this virus is not as virulent, so it's not as severe for people to, sorry for my cat in the background. It's like, So if it's doubling at this pace, well, you might end up you will end up having 1 million people. But even if it's less virulent, you might still overwhelm the healthcare system, because if you have severe cases with such large numbers, just just because even if it's less virulent, the absolute number of patients that end up in the hospital is going to be large enough that you basically cause problems for the healthcare system.
Sorry, Cheryl, if I may ask, somebody in the chat is saying 1 million new cases a day in the UK or in the world?
That's in the UK alone.
Okay. So in the world, what would you say?
Well, that depends on the speed it spreads. So you would actually have to calculate that specifically for the world, because as it transmits to the world, every single place is a little bit different. So there are you cannot generalize everywhere. For example, there was a strain called the Mu strain. Mu appeared in Colombia and overtook everything else, but in worldwide, it didn't spread very much beyond South America. But you cannot extrapolate it. The data is very specific to places and things like that, but it should be something, faster than Delta for sure. So I would say that that's a safe statement. They say that it would be faster than Delta in pretty much every setting.
Mm-hmm.
So okay. So if you go, if you look at South Africa again, this is then Gauteng. If you actually look the Omicron wave, you can see if you actually compare to the Delta wave, and as you can see the cases and admissions and death is always a lagging indicator. It lags a few days behind. So that's you have if you actually look in South Africa, the number of cases and admissions are like trackly close together. But that's because of the South African healthcare setting, where people only go in when they actually need to go to the hospital. They don't go to get checked as regularly as as in other places. But if you actually see the number of deaths and hospital admissions is decreased. But as I said before, you have to consider that almost everybody has been infected before by a previous coronavirus wave. And it's you have the less deaths and less hospital admissions, maybe because you have two things that you're observing. One is that you might have pre-existing immunity, but also that you may that we're seeing a younger population coming in. So that is maybe because they were not infected before or something like that. We don't understand it very well, but this is the observation. So by the fact that you have a younger person coming into the hospital, it's severe to go to the hospital, but you will probably not end up in the ICU just by the fact that you're younger. So this may not necessarily mean that a person who is naive, in the sense that has not had COVID and has not been vaccinated, maybe in that case could actually be more severe, not less severe. The history of infection affects the outcome severely, and the age of the person affects the outcome severely. So, we cannot say that at this point that the virus is for sure milder for an immunologically naive person. So we cannot compare to the initial Wuhan situation very easily. And this is basically what they saw. So this paper was like amazingly quick. This appeared on December 1st, which was basically less than a week after the virus was officially publicized. And what you actually see here is very, very nice work, given the amount of information that we have. So they actually use a very, very clever way to estimate the immunological escape due to Omicron. So what you actually look at is basically like, how many days did it take for a person to be reinfected after every wave? So if you look at the people, so basically like with beta, it took over a year for people to be reinfected on average. That's the purple wave. With Delta, it took just under a year for people the majority of people to infected. But with Omicron, people who were previously infected are getting reinfected, as fast as 90 days. So basically the previous infection, this is basically showing that the previous infection by SARS coronavirus does not seem to give people protection. Whereas before we saw about 80% protection when people got infected by by SARS-CoV-2 and had COVID. Very quickly after that, people in laboratory work, they showed that basically vaccine serum and monoclonal antibodies, the commercial drugs that are basically they're based on antibodies, are not very good at neutralizing Omicron. You have decrease on, most people are showing between a 25 to 40-fold decrease in basically neutralization. And typically, for example, for flu, when we have a 6 to 7-fold decrease in neutralization is when we say that we need to update the vaccine. So it's time for us to update the vaccine. And if you actually look at, people actually see that breakthrough infections with SARS-CoV-2 Omicron, even when people have boosters, people can be infected. So this is basically proof that this can happen. But now it's like comes the question like, how frequently does it happen? Does the booster actually offer you protection? And the answer is yes. So back to the UK study, they check basically how people do with booster compared to not having a booster. And, just after booster, people, with a fresh booster, people can see 70 to 75% protection. So you remember when we actually had with Pfizer or Moderna, we had something around, 94% protection in the clinical trials, and probably like 92% in terms of in the real world after the clinical trials, what people were observing initially within the first 3 months or so. Here we actually see decrease to 75%. So it's basically like now it's working about as well as the AstraZeneca vaccine. So, and just to make this real, so there was a party in Oslo, and as you can see that, they had about 100 people at a restaurant, and 60 of those people, about 70% of the people, 60 of the people who were visit the restaurant were infected. And basically you had 111 participants, 80 people were diagnosed with SARS-CoV-2. And so far, like 70 of them have been confirmed by Omicron. It's just because not everybody has been sequenced. So it's very likely that everybody at that party was infected by Omicron and not something else. And the interesting thing is like everybody in this party had to be vaccinated, at least with 2 doses. And they were all checked by antigen tests. So it's basically that you just have to have one escape. Just remember, antigen tests are very good for people who are symptomatic, but they're not very good for people who are asymptomatic. So, this is the best thing that we can do, but this is not, so anyway, so this is basically that they could confirm the vast majority of people had like 2 doses of mRNA vaccine. So this was supposed to be, so just remember that, after 5 or 6 months, you're not going to have much antibody. The antibody is undetectable against Delta or Beta, for example. So here's basically like, so according to the company, the like only vaccinated staff had been to the party, and everybody had tested negative for coronavirus for the event. So they tested everybody, yet the virus was able to infect all these people. And this is from the New York Times report. Here, this is not an isolated case. So in Denmark, there was another Christmas party where 53 out of 150 people were infected. And then there was a 60th party at King's College where 14 out of 18 people were infected. So, and basically everybody was vaccinated, and they had like another, everybody was vaccinated, and and basically everybody had basically antigen COVID tests, which is basically like the little sticks that you have where you actually try to see like 2 dots or 2 lines that to test for a virus. So, you know, you only have to have one escape, right? You only have to have one escape or one antigen test, and not all antigen tests are of equal quality, as some recent publications have shown. So some are better than others. So you have to keep that in mind. So, you know, how much more transmissible this is, what came out in this case report of basically in a quarantine hotel in Hong Kong. So in the quarantine hotel in Hong Kong is basically you had travelers that came at different days, but one infected from one room to the other room. And the only time, you know, from video surveillance and, you know, in the hallway, people figured out that there was no contact between those 2 rooms. However, you know, they had to serve the meals, and they had opened the door and then closed the door basically to serve the meal in one room. And after the closing the door, within 1 minute, the second door had opened. So across the hallway, within 1 minute of basically of air left in the hallway was sufficient to transmit to the second room. So this is just gives you an idea of this enhanced transmissibility that we're observing here. And now talking about this, given this level of transmissibility, then if you look at, this is the European CDC, you can look at, you know, the number of of cases across Europe on December 10th. And remember, the travel bans were imposed, you know, they were imposed immediately within 48 hours of the virus being announced to the world. So, you know, this is, you know, these are the ones that you found. You don't know which ones you didn't find, but given the, you know, given the spread and the speed at which it spreads, we saw immediately, for example, in Scotland, you know, very quickly, you know, basically community transmission. Same thing was with Denmark. All these places that sequence a lot, you're finding, you know, large amounts of transmission. So, you know, the take-home lesson is that border controls and these flight restrictions are mostly ineffective. All that you do is like maybe you get a couple days delayed. I mean, the only way this kind of border control things will work is like if you do it like China does, which has basically typically a 21 or 28 day, you know, complete quarantine in quarantine hotels with multiple tests, et cetera, et cetera, et cetera. But, you know, anything less than that will certainly be insufficient to stop this. I mean, these quarantines are not completely harmless, as you may know, because if you ban flights, so one thing that has happened now, the South African health authorities are running out of reagents to test for the virus and to sequence the virus, because these flights are not only carrying passengers, but also carry cargo. You know, most of the planes nowadays carry a mix of passenger and cargo. And now they're not getting their reagents to continue monitoring and finding new strains due to the travel bans, because the airlines are canceling flights. So these things have to also be considered, not just, you know, the passengers themselves. So, well, the good news is that, you know, the good news is basically that we actually had boosters, and I talk about this in May. So in May, I just call for, like this says, we need to have boosters, because, you know, with the African and so basically the South African B variant and the Brazilian P1 that we had at a certain moment, you know, 6 to 8 months after completing vaccinations, 50% of people had no detectable antibodies against those variants. So basically they were not working at all. So I think 6 months is a reasonable time to have a booster. So I think, like, you know, we should actually try to get everybody, you know, have a booster. But the boosters do work. So we actually see that, from the data in Israel, where they actually boosted very quickly as soon as they found that the immunity waning, you decrease the lethality by 90% due to COVID with a booster. So, booster is definitely beneficial. And if you actually look at the protection from the booster, you can say very, very quickly the curves start diverging. So this is basically the number of people who are infected. These are the people without booster getting reinfected. This is breakthrough, what we consider breakthrough infections of the second dose. If you actually add a booster, the third dose, you flatten it out for quite some time. And this is monitored over, over 3 months. So after 3 months, it's still working really well. So you actually decrease the infections, the breakthrough infections, a lot by having a booster. But, going forward, I mean, people are complaining, is like, how come we had, everybody has the impression we had smallpox vaccines or polio vaccine that everybody has, and then you have immunity for 40 years. And then like smallpox vaccine, or use vaccinia, you have, immunity for 75 years. And this is from Fields Virology, which is the most standard textbook in virology at, at a graduate school level, or even undergraduate level. We've known forever that immunity doesn't last very long for mucosal infections. And mucosal infections are basically like influenza, RSV, the things that infect your airways and your lung and your gut. Those are mucosal tissues. So infections that happen in those tissues, they don't last very long. So an RSV is as short as 3 months. The immunity only lasts for 3 months. So why do we expect, why do we expect the immunity to last forever with the coronavirus vaccine? I really don't understand. People just didn't read the textbook or something like that, or people are talking out of their area expertise or forgot about this kind of thing. But I mean, everybody who knows like mucosal virology and viral immunology, knows this, that is like, there is no expectation to have this this thing forever. It's not just this textbook, but other papers, even early on, in professional circles. See, this is like May 22nd, 2020. And this is basically projecting the transmission of SARS-CoV-2 through the post-pandemic period. Once everybody is infected or vaccinated, what's going to happen? So this is based on the other coronaviruses that we call them so-called cold coronaviruses. So focus on this. This is the real data, or the solid lines. But if you actually look, it always goes through these yearly cycles. So this is how coronaviruses work. So we should expect this to come periodically, because unlike flu evolves by evolving faster than your immune system. So the immunity to flu lasts about 3 years, as I showed here. Is an influenza here. It's about 30 months. But coronavirus is only 12 months. So what happens? So then it's basically you get an infection, your immune system acquires memory, but over that year, then it loses the memory and can be reinfected. And what happens is, why do we have these mucosal things, mucosal infections, lack of memory? So this is very simple. Your tissue turns over, and these resident memory cells sit in between your basically mucosal cells and your lung epithelium, or your throat, or your gut. And the cells that are called resident memory cells, when the tissue turns over, because these tissues turn over quickly, the memory cells get released and migrate back to the bone marrow. And when they migrate back to the bone marrow, they forget their immune memory. So that's not going to last forever, just because these tissues. So this is intrinsically the biology of humans. This is intrinsically the biology of the virus. So this is the way the virus is successful and can come back every year. So it's going to be very difficult to tweak the human biology, as well as the virus biology. So I don't think that any vaccine can go beyond a year. So that's my opinion. And that's based on, what I've shown you here and the body of evidence that's behind that. Also, we'll never get rid of the virus. So the reason I don't think we're going to get rid of the virus is because we have so many animal reservoirs. We have, it can infect humans that we care about, but it can infect dogs, cats, lions, all the mink that got culled early on in Denmark that was actually generating new variants. Like the wild mice in North America, like Pyramidus species, they're a susceptible host also. They can transmit it. There's a paper that came out just recently, also the end of last month, that shows that 40% of wild deer, this is white-tailed deer in some of the Midwestern states in the United States, are positive for the virus. So, given that you actually have all these animals being infected and it can spill back onto humans, there is no possibility for you to get rid of the virus anymore at this point. I mean, not to mention the original bats that had them. So you cannot prevent that from happening. So all that we can do is surveillance and manage as we go. But if we check as managers, so this was from a talk given by the chief scientific officer of Moderna. And the good news about this is, it only takes 2 days to redesign the vaccine. So you can actually update the vaccine in 2 days, to redesign it and have the new updated for the particular variant. And then to retool the entire manufacturing process, it takes basically like 42 days. So from 45 days, you can actually have a retooled manufacturing. What takes a long time typically is the clinical trial. But, that is basically the clinical trial that we need to have here at this point is not like what we had last time, but we need to have a clinical trial that is similar to what we do for the flu virus, which we only check for safety, not for efficacy, because otherwise we're not going to be able to move fast enough to vaccinate faster than the virus can come up with variants. So basically you test the vaccine for safety, but not for effectiveness. And you assume it's going to work as well for effectiveness because you are actually giving it exactly. You just have to make sure that whatever you're injecting is not dangerous. If we move to this flu model, which is what we're doing already many years, that is the way that we can keep up with the virus. So anyway, so then it's like here, here we actually have basically sort of a summary. So the good news is that we know how to manage the virus by, reducing risk. The booster works, at a 70-ish% level. How long it lasts, we're not sure. There seems to be less hospitalizations. We don't know exactly the reason why there's less, but that's what we seem to see. But if we have absolute numbers that are really high because of the high transmissibility, it can still overwhelm the healthcare system. So we have to be careful about that. And anyway, the other thing, the thing is, the bottom line is that virus spreads much faster, and the spread, we don't know exactly how much faster, but it's significantly faster than Delta. So that was the fastest one that we ever saw. And, we can see probably something between 3 and 8-fold faster than Delta. So it's very significant. So anyway, so with that, I'll put up my acknowledgements and also, take questions.
Super, Gerard. Thank you so much as always. And you have already 26 questions waiting for you, but perhaps I will ask the first one that I can see, and I'm sure it's everybody's minds, is knowing all this, would you suggest traveling by plane home to visit our elderly parents over Christmas?
Well, you saw the numbers that I put, the projections. So hold on, let me just put it up here. So I would say there are two ways to go about this. One is to get ahead of the virus. And then, and depends on the local situation. But this is what worry me, because it's like, it says that by December 25th, I have 1 million cases in the UK. I don't know how many cases you are. So I think is that, I would say if I were to travel, I would travel up to this week or beyond. But I mean, I would say that because this is a projection. This is basically you just fit a straight line and your estimation is based on fitting this straight line. But if it's actually slower than that, then I would say that, it's probably okay, for you to visit your parents. It's kind of reduce risk. And also depends whether your parents are boosted. So if your parents are boosted, recently, I would say within less than 3 months, I would say, and about maybe a week after, I think is a reasonable risk to take. That's what I would do. I mean, I went to visit my aunt in Vancouver. And just before that, like a month before, I took a booster and then I went to travel. That was basically in November. So that's basically I would do as a responsible person. And then, just before meeting up with them, I would take an antigen test as close as possible. Being fully aware that it's not a perfect measure, but since you're traveling, you're also going to take a PCR test. So a PCR test, you'll take, and then within, after you land and go and wherever you may go, you may want to take an antigen test before going to visit your elderly parents. But the best thing that you could possibly do is you being boosted and your parents being boosted. And I would say within 3 months, if it's within 1 month, is even better. Your peak immunity is going to be about a week to a month after that booster that you get. So that's how I would do it.
Super. Our colleague is saying, thank you so much for the tip. I'm sure that is helping a lot of people. So thanks for that. Another question is, does Omicron cause or could potentially cause more severe disease, and could it be more dangerous from this point of view?
So as I mentioned before, I think Omicron can possibly cause more severe disease to a naive person. So a person who has not had a previous case of COVID and has not been vaccinated. But in the context that we're seeing, either in South Africa of people who are natural infections, and what we see in Europe of people who are who are already vaccinated, we see milder disease. But that doesn't mean that there isn't more severe disease in the case of a person who has never been never developed any kind of immunity for whatever reason. So that is what we cannot treat everybody equally. It doesn't work the same. It depends on your history of infection as well as immunization. And there's something called affinity maturation. So when you see the same antigen, like the virus, more than one time, your antibodies actually become better over time. So that's another thing. That's like if you see it more times, it actually it can become better. So that's another reason why we think that over time the immunity actually becomes better.
Mm-hmm. So another person is asking, is it so that all vaccines need to be adapted because of Omicron, as they seem not to be effective enough? So you were telling us that it can take, a couple of days to adapt and the process a bit longer. But so do we have to adapt all the vaccines that we know?
Yes, we have to. I mean, this is with pretty much, with things like flu, we are doing it constantly. Every season we get a different flu vaccine, and it's a guess. So people normally monitor in China, where people have a lot of ducks, and they have basically the largest bird population in the world, and people monitor there. And then based on what appears there, they guess what next year is going to show up in Europe and North America and South America, because the virus spreads in that direction. So that's what people typically do. So that 6 months advantage that you have is basically you take your best guess, you manufacture it, and then distribute it, 6 months ahead of time. So
Yeah,
we have a
so
cycle like that. So you have to adapt the virus, adapt to the virus. In this case, and basically coronavirus will be a little bit different. If we had, done it back then, this is when we actually saw the original Wuhan virus, and then we says, aha, the UK variant is starting to show up. We're going to adapt it to Alpha. And then when you saw like basically, when you saw Delta coming up, then you would adapt it to Delta. And this actually could go from us having vaccines that last like basically 6 months, maybe it can push us to close to a year, with some other technological advances that will be necessary. But I think that's the way to go forward.
So now if we have been given the booster shot, that was probably have not been adapted for the Omicron variant. So to what extent, you were saying it goes down to 70% then of protection, or how does it go?
Well, right now with the original Wuhan virus, we're still getting 70% protection. But if we were to take even Alpha or even Delta that have more mutations that are similar to Omicron, will probably be better, almost certainly better. So in the flu case, we take not just one, but we take a mix of our best guesses. So you don't take just one, but you think, okay, which are our best guesses, and you pick like 3 or 4 of them, and then you mix them, and then have vaccine that contains basically maybe a couple of variants. The thing is, like, it doesn't have to be exactly Omicron. It just has to be close enough. And then it's basically, but now Omicron is basically like 52 mutations away from the original one. But if we were at, at the Delta, or we would actually have fewer, because these mutations, many of the mutations that we see are mutations we've seen before in other variants. It's just that this one combined them all. So some of them are unique, but the majority we have seen before. So yeah, we have a sort of a general idea of the kind of things. I think over time, as we see more, we can see if the virus actually limited in the types, on the number of amino acid changes and type of mutations they can have. And then we can probably design better vaccines over time, and basically delineate the space in which the virus can mutate. And then we give them the basically design vaccines within that space that will actually, it'll give us the ability to guess better what the virus will do next.
That is kind of the question that we're getting now, is will we see the same wave of propagation worldwide compared to the previous years with the previous variant? In other words, would there be a way to foresee its impact? Could we do a model in terms of public health policy and further measures to take? So can we project, can we make a model? And can we, as you say, predict the space and do something for next year's?
I mean, people do generate models of such things, but they tend to be difficult, and they tend to be relatively short term. And as you see in the picture now that I have below here, this is a typical model, and this typical model is just a straight line. But if it actually, models are more sophisticated, or have curves and things like that, there are different ways to model things, and that's why it's important to model things, also have more sophisticated models. I mean, the first model that we all do is kind of like this, but if this goes exactly like the way it has been going the last few weeks, how does it go, and how does it, what do we project to coming forward? Everybody knows that this is not a perfect model. This is a very rough estimate, but this is not, but this is the first thing that we do. It is called like linear models, which is because they're straight lines. So that's basically like, if we increase every day by so and so much, what we're going to get. Of course, it's not going to work like this, because, I mean, if anything, you eventually run out of people, then you flatten out and things like that. But anyway, that's that. Yeah, we do, many places do modeling, and I think it's like public health modeling, and that's their business. So there are people who do this. The problem is like, I don't think we have many models that are society-wide to every aspect, or basically take into account all economic aspects, all travel aspects, et cetera, et cetera, et cetera. They're all partial models. We don't have many of those. And that goes into another area that I work in, basically like big scale modeling and big data that you actually use. If you were to integrate it, now we do have computational capacity to do these kind of things, but it requires people who are dedicated to integrate all this data in such way, which is also not easy.
Mhm. And you were saying before that we should adapt all the vaccines to the new variants. This is what we were discussing before. But what about the treatment that you give once you're infected? Should that also have to be adapted because of the new variants?
Well, yeah. So I think it's like there's a in that area is like the two new things that have appeared. So before, I think it's like many treatments have been mentioned, and now with maybe 2 years hindsight, we kind of have a good idea of what works and what doesn't work. So most of the treatments that we have that we mentioned before, they essentially don't work. So for example, I would say that things like antibodies are the only things that really work, you know. So the new things that seem to work are Paxlovid and molnupiravir. So molnupiravir is basically a nucleotide or nucleoside. That is basically a piece that is basically something that looks like a component of RNA. And that is basically targets the viral polymerase, the enzyme that the virus makes copies of itself. The other one is Paxlovid, is a protease inhibitor. So the virus when it makes initially its 1A gene. Let me go back to the very first. So it makes these large genes here, but it has to cut them up. So we see here in between this, the beginning of the end of the genes. So this has to be cut up by something called a protease, and it has to cut this up into pieces that the virus can actually use. And in order to do that, it uses this protease enzyme, and this is this Paxlovid is basically an inhibitor of that. We actually use protease inhibitors against HIV, but it also we use nucleotides. The thing is that most of the time, if you just use a single one, the virus eventually becomes resistant and it doesn't work well. And in the end, for example, with HIV, we had to use combination chemotherapy when we actually had to combine them. But right now is like, you know, these two, one is made by Merck, the other one is by Pfizer. So then people would actually have to come with an agreement, and basically they have, each has their own interest. But in the end, I think we need to combine them in order to prevent resistance to these drugs. And the more worrisome drug, some people have worried about molnupiravir, because molnupiravir works by an interesting way. It's not a drug that inhibits the enzyme in the real sense. So it basically, it doesn't stop the viral enzyme from working. But what it does is like, it makes the viral enzyme make many mistakes. It makes so many mistakes copying its genome that it can no longer survive. But if you had, for example, low adherence, you make mutations, but not enough to kill the virus, then potentially you could actually allow the virus to make more mutations and make more variants more quickly. So there are people who worry that if you have poor adherence and people don't take the drug as they're supposed to, or they misuse the drug in some way, let's say not giving the full dose or not finishing off the virus completely before stopping, like the same we have antibiotic resistance, we could promote the generation of more variants. So there are people worried about molnupiravir, especially if you don't combine it with Paxlovid. So, and the other thing is like all these antivirals, they typically only work early on in the disease. So ideally you would actually, let's say, test positive. As soon as you test positive, you get on this thing, and then you get on it for maybe a week or maybe 2. We have to see how long this actually takes, or it could be as short as 5 days until you absolutely see no more virus in the person, and then you stop. That would be the correct way to use it. as far as we can learn from previous experience with other viruses.
But would you need to change these ones then with a variant of Omicron, for example? Is there?
It doesn't seem to be. So the areas where you actually mutations are not the areas that are basically the genes that are the protease and the RNA dependent RNA polymerase. These are not the places where we actually see the mutations that allows us to expect that allows to expect that these that will be changed by Omicron. So most of the Omicron, the mutations are happening in the spike. So there are even some. I think there were just yesterday, there was a study that came out that I think addressed specifically, I think for molnupiravir, that said that is like, I haven't read it yet, but I saw the title and the headline that is like that, that doesn't seem to be affected.
Is there a level of vaccination which would prevent the emergence of further variants like Omicron?
I think in a practical sense, it's not going to happen because first, like you have to, with this level of infectivity, the percentage of people that you would have to vaccinate would have to increase. In addition, you do not only, it's not basically like you have to vaccinate everybody, or like I would say like 90% of the population probably with this level. With measles, we have to infect like 97% of the population or something like that. If you have to infect that many people, and it has to be done within 6 months, everybody. And we know there's breakthrough within, with Delta, for example, within 3 to 4 months. So you would actually have to vaccinate the vast majority of the world at 90% level within 3 to 4 months probably. So I don't think that's going to happen.
And what is the main barrier to that?
Well, first is like vaccine production capacity. That's one thing. The other one is delivery. So for example, from smallpox, it's like reaching everybody everywhere. It's going to be, but this is never going to remove the virus altogether because you're going to have like spillover from wildlife, your reservoir. So now that this thing is so widespread, humans have spread this all over the world in all kinds of animals that we don't even know of probably. Now they can actually bring it back once our immunity wanes. And it's going to be so difficult to keep it up with the majority of the world. We have to manage to a reasonable level at this point. We're never going to be able to get rid of it. We just have to have an infrastructure that allows us to adapt and change as fast as the virus itself. So this is basically like going to, what Richard Dawkins called the Red Queen hypothesis, where, in an evolutionary race, you have to run as fast as possible to stay in the same place.
When you mentioned that the doubling rate depends on the environment, what do you mean by environment? What is it made of? Mainly collective immunity, or is there some other elements to take into account?
Many things. There are going to be many things. So there's everything that affects the virus spread and things like that. The environment could include, as like the immunological memory of that, the immunological history of that population. It could be like the density of that population, how people move, where there's like, for example, a population with a lot of commuting, air travel, those are the things that we're trying to change by, let's say, working at home, putting masks on. That's part of the environment too. So the environment is basically every possible factor that could affect the virus that is from the environment, like how much people aerate things. Those are all the environmental factors that affect the spread of the virus. So also the antivirals that we might be using that put pressure for the virus to develop resistance against, is basically everything.
Another question here: can it happen that people who had just received the booster vaccine are infected by COVID? How this could happen? So how can you get infected when you have got the booster?
Well, people can be, as I said before, that booster is not 100% protective. Each person's immune system is a little bit different. So for example, you can already tell from experience by talking to your friends, some people got basically a side effect and were sick for typically like 36 hours. Other people didn't feel anything. So that already tells you that that's the immune response of every person. So, you know, and also, for example, young people have typically a very strong immune side effect profile, especially, you know, in the late teens to early 20s is like really, really bad. Whereas like you rarely have any 70-year-olds experiencing side effects. So that tells you like the immune system is different. It's reacting differently for, you know, so that's when we look at the, you know, so not everybody's immune. The other thing is like with this one, with Omicron, it only gives you 70% protection. That tells you that all the other people, you know, are actually getting infected even when they are boosted. So, I mean, there is no way around it because that's basically the best that we can do, the best that we can offer as a society. And then this is a part of your biology and also of the virus's biology. And that's the outcome based on what you're given. It's like that what you're born with and with your immunological history.
So basically, it's kind of a lottery, you know, for each one of us. We don't really know what will happen until it happens.
Yep, exactly. But we can be pretty sure that at the very beginning we are, especially with this. But if you actually had, let's say, if you actually had, for example, a booster for, let's say, specifically for Omicron, when Omicron shows up, I would expect that, you know, the effectiveness would go back to about 95%. So then you would be, instead of being protected 70%, you will be protected 95% against breakthrough. So especially for the first, you know, 3 or 4 months.
But of course, for those who have been, you know, got the booster right now, with the booster not being adapted to Omicron, they're down to 70%. And if we see, as you were saying, that there's a very strong spread, a very, very fast spread, well, that means that we're kind of a difficult situation in these days coming up to Christmas.
Yes, there is no doubt about that. Yeah, there's like, it's going to be, yeah, we were, as we have seen previously in the last 2 years, again, caught a little bit off guard. At least our surveillance is good, especially in Denmark, in the UK. Denmark is sequencing about 10% of cases. You know, the UK is sequencing at least 5% of it. I mean, those are very good levels, but I think it's like, if everybody else did the same thing, I think we'd be better off. You know, it's always good to know because then you can do something about it. If you don't know anything about something, there's nothing you can do about it.
Okay, and you were just mentioning before that the side effects that we have seen from the booster shots in young people are stronger than in older people over than 70. So can you say more about the side effects? What sort of side effects are they and how dangerous are they for the young in case of the virus?
Well, it's like there is, so the side effects are I would say uncomfortable, but they're not dangerous in its majority. The only problem in like younger males is that you actually see some cases of myocarditis. It's not exactly clear why this is. So myocarditis is basically inflammation of the heart. In most cases, this also resolves. But there is some thinking that this could be due to vaccine leaking out and somehow getting to the heart. So then the logical thing is like, maybe is these young people are like exercising or doing something with their deltoid that is basically, or doing something, you know, active that led to the vaccine to leak out after being injected. So there's some thinking of that, although this is not proven. This is sort of basically people have hypothesized this is the case. So maybe, to prevent that, maybe don't do anything for a day or so after you're vaccinated with that arm, just in case this is right. But I cannot say this is for sure, but that some people have thought that this might be the reason, but it's most likely people think it's related to the vaccine leaking out of your arm. Basically,
what about something that is worrying a lot of colleagues, all this controversy about giving it to 5 to 10-year-olds? What is your view on that, giving vaccines to 5 to 10-year-olds?
I think it's like, in the end, we should get children vaccinated. The children, I mean, there is, in young people, there seems to be long COVID also. Although, they might not experience things immediately, but so and also as a society, to protect everybody else, we should, get as many people vaccinated as possible, because you also stop the spread. So you do not, as a society as a whole, but for example, let's say, okay, somebody may think, okay, it's not worth getting the risk of to get my child vaccinated because like that. But what if you actually end up the child bringing it to the parents, and the father was, let's say, for example, or the mother, who's the breadwinner, gets severely sick and then can no longer provide to the child? What are you ultimately benefiting there? So I think we should not think things as simplistically, and we should think things a little bit holistically. What does this do as a whole? And not only that, as just directly to the parents, but as a society in general. If we had this population of small children who are spreading this virus all over society and eventually killing your grandparents, what does this mean? How do we affect society that way? So I think it's like we have to think things a little bit more holistically rather than just as individuals. Although we all value our individual freedoms, I think there's also time that we have to take like social responsibility for society at large. And I think that's some of the things that need to be considered. But for the children themselves, some of the, although not common, you do have COVID related problems that, that are very significant. Like for example, some inflammatory syndromes that we actually have seen.
Yeah, but you were saying that in terms of vaccines, I know that there hasn't been a lot of tests on children, so it's difficult to derive or extrapolate any conclusions, but are there any reasons to be concerned to give the vaccine to children? I mean, the vaccination itself?
No, actually, there are, I mean, the anything that is, I mean, the vaccine clinical trials have been done, and the thing that has been looked at mainly in the vaccine for in the clinical trials for children was toxicity. So basically that's exactly what has been focused, and all the approvals have been, the only reason it's approved is because it passed their clinical trials, and the clinical trials they evaluated did not see significant toxicity. So that's, I mean, that was the safety was basically the number one priority in clinical trials. And that's basically, so that's why I would be completely confident to give it to a 5-year-old.
So when you say toxicity, you mean that in general, any sort of consequences because of the vaccine on the health
Exactly,
of the child?
yeah. So basically the vaccine shows no evidence of, of safety for children. Okay. Up to 5, that's what has been done so far.
Okay, great. And what about another controversy that we have heard a lot lately is that the crossover of vaccines. So for example, you have 2 doses of Pfizer and then you get your booster.
Yeah, so there's a nice study in Lancet that basically showed all the combinations, but basically all the combinations work. And the combinations frequently work better than just the single company vaccine. So yeah, there's a paper that showed that in Lancet. I didn't put it up, but yeah, that has been done.
So that it works better, for example, 2 Pfizer and 1 Moderna as a booster. In theory, it works better.
In general, and also there are several papers that show Moderna is a little bit better than Pfizer. But that's I think in a practical sense, it probably doesn't make that big of a difference. But for example, it is about twice as good as Moderna is about twice as good as Pfizer for breakthroughs.
Mhm.
So
And in terms of the symptoms when you get the vaccine or the booster, some people have stronger kind of.
so yeah, so
how do you
Moderna
explain
has
that?
stronger side effects than Pfizer from what I've seen, and yeah, so that seems to be also, but the dose of Moderna is about over 100 micrograms versus the Pfizer one is 30 micrograms. So of course it's, significant, 3 times more. So that could be the simple reason why this is the case.
Okay, but not to get worried for our colleagues, no? So if they have got a strong reaction, it's just going to be temporary, and it's
Yeah,
showing that
it's
works.
basically like the onset is about 12 hours after your vaccine, and then it lasts for about 36 hours. And for most people that this is anecdotal, and and I've talked to a number of primary healthcare providers, and they seem to report also that the booster is milder for people than the second. So the second, people had the strongest reactions, and then the booster people seem to be, generally speaking, milder than the people who have experienced a stronger side effect. Has been like, for example, people who took Pfizer, the first two, and then Moderna, which is itself stronger, then experience stronger side effects with Moderna. But
Mhm.
this is all within expectation.
Yeah, and now a question about the time when you get the booster. Somebody's talking about boosters. We've been told that we should wait a certain period of time before getting it. So what could happen to people who will have their boosters in the first trimester only of next year, given the fact of the doubling rate of Omicron?
Well, I would say that, if you were to ask me my opinion, I mean, typically the scheduling of boosters, I would say that for sure every 6 months you should get it. In the case of Omicron, I would feel confident that if you take like 3 months after your second shot, would be okay for you to get a booster. But that's going to depend on your particular country's health authorities, whether they let you or don't let you.
Mm-hmm.
So you might not have much of a choice there.
Okay. So based on your experience studying previous strands, what do you think would be the best policy mix to fight and eradicate the virus? Many people are beginning to lose trust in the government and institutions, as previous lockdowns and other measures, social distancing, masks, were not that effective. So, I know you have been answering this question in several ways, but the question really is, what is the best policy mix to fight the virus?
I think the best policy mix would be, that you vaccinate the population as much as possible every 6 months. I think it's like, if we actually have that with basically on top of that with a solid monitoring, basically based on the things that we actually have now, just with a solid monitoring infrastructure, that you can basically adapt the vaccines and have it basically people routinely be able to take off 2 days every 6 months in order to get vaccinated and deal with the side effects, that you can basically have extra sick days that you can actually have. I think that's, a good compromise. I believe that if you were to do that, society wide, and there would be good compliance, I think we could be pretty much normal, like in the times that, but basically I don't think we're ever going to go back to where the virus is eradicated. As I said, I don't think that is possible. At best, we maintain it at bay in the animal reservoir, and occasionally we have spillovers, but I don't think it'll be possible to permanently eliminate. The more we suppress the virus by vaccination and keep it only in the animal reservoir, the rarer variants are going to be. Instead of having, basically 1 billion people in the world playing the genetic lottery for new variants, if you keep it down, then the time between variants is going to become longer and longer until you basically maybe you don't even see it within a generation. So, but that is possible. But the question is whether we as a society can change ourselves and make this into a routine. So getting vaccinated every 6 months, have a solid, monitoring system, and also allowing the technology to go forward. Because right now I'm telling you based on the things that we have now, but if you need to have research on the virus itself, you might come up with another trick to fight the virus that you didn't have before. And we just don't know what we don't know, right? So that's what research is trying to reduce, the stuff that we don't know. And the more you know, the more ideas you can get about how to do something about it. So I think you will actually have to have those 3 lines. So.
And you were saying before that we need to, we need to see also the profile of the people that are, for example, in hospitals now. So that's one question here is, do you have some clear idea of what is the profile of those people that are hospitalized because of Omicron? So any particular profile?
Well, all that we know right now, because the numbers are still relatively slow and low, and then is like what the Omicron profiles seem to be. First, the cases are milder, but they're also younger. But they also have a history of being previously either vaccinated or infected. So before, the people who are convalescent would just not show up in the hospital. But now we have, and also this appearance of school children and college students. So to show up in the hospital is quite rare. So that speaks also to the case of vaccinated children are 5, because now they're showing up in the hospital with Omicron. So I think that is a good idea to, get some protection.
And what about, it seems in many countries we have an issue with the anti-vaxxers, so those who do not want to have a vaccination at all, and it seems to have kind of impacted on the different policies that they have put forward, the different governments. What to do with those cases, and are they being affected more or more virulently than those who are being vaccinated?
I don't know, but the expectation would be yes. That, I mean, the thing is, like, you will be immune at one point, whether it be by natural infection or by vaccine. Eventually, I think everybody will be immune, but the consequences of being, from a natural infection could be that you have a 1 to 2% chance of dying versus or having long COVID, which also seems to be increased compared to the people who are vaccinated. So it's up to you how you want to achieve it in the end.
But is it safe to say that most of the people that are in intensive care are non-vaccinated, as we have been hearing often?
Yes, at least for Delta. Basically, a Delta, at least in the United States, has been basically a wave of the unvaccinated. So somehow, even when people surround themselves with vaccinated people, so on, or live in isolated areas and like low density states, eventually the virus somehow gets to you. It's like the virus will find you if you're unvaccinated. That seems to be the take-home lesson of the last half a year or so with Delta. So yeah, I think that's going to be the same everywhere. I don't think that's intrinsically. The United States already, compared to Europe, has much lower density. And distant states like Maine, where you actually have high take-home vaccination, we have over 70% of the people vaccinated. You still see these waves coming of people are basically who are unvaccinated, who eventually end up sick and in the hospital. So yeah.
But what is the proportion of those people? Because the point I'm trying to make is it a clear conclusion that if you're not vaccinated and you get the virus, you're going to have much worse consequences, and the percentage of people in intensive care is higher?
That is clearly the case. It's like, I think it's like, I was talking to some primary care person, and they said, like, they only saw one person who was a breakthrough, who had basically underlying health issues, who was immunosuppressed, was careful immunosuppressed, but it's like had another 35 people in the hospital who were not vaccinated. only one person who had been vaccinated, and that person already had, this is just in Los Angeles, who had underlying health issues.
So, I mean, that's a very important message perhaps to end, because we're overstepping our time, but
Yeah.
you are such an amazing source of information that we are abusing you a little bit. So can we say as a conclusion that we're all going to get infected one way or another, from what I'm hearing? It's up to us to see how we want to face this. So we can actually, and your solution is, or your suggestion is, to be getting boosters in a regular way every 6 months, if possible, as many people as we can. And that if you don't get those boosters, that the chances, or that vaccine, chances are that you're going to get infected almost for sure. And that we should also responsibly not just take care of ourselves, but of other people, and that goes particularly for the kids and vaccination of children. Could we say that is a safe assumption, all the things I've just said?
Yeah, that's correct. That would be the correct take-home lesson.
Okay, well, thank you so much, Gerald. As always, it has been such an amazing pleasure. I am so sorry for the 64 colleagues that have been asking for questions that we haven't been able to put forward. But of course, I didn't want to interrupt the flow of your answers, because I think that every word counts. And of course, as always, talking about abuse, can we abuse you again some other time to see, perhaps after the holidays, to see how things are going? Would it be okay for you to come back?
Sure, we can do this again sometime.
Super. So thank you so much, Gerald. And I profit to say Merry Christmas to you, to everyone who's listening. Merry Christmas and safe Christmas, because we want to see you all back again for our next wonderful event with Gerald. Thanks again. And please, Karen, if you can end the session.
Happy holidays to everybody.
Thank you.
Thank you, Anna. Thank you, Gerald. I'll just stop the recording first of all.